Hello students, today we are going to, in this video of ours, we are going to learn about the process of gel electrophoresis. Now if you remember, we have done this gel electrophoresis while we were doing DNA fingerprinting. While DNA fingerprinting also, we are using this gel electrophoresis. Gel electrophoresis is the process by which we are isolating, we are separating the DNA fragments because we want the gene of interest. We want the fragment of the DNA where our gene of interest is there. So once we have taken out the DNA, we have to separate it. And for separating this DNA, the process which is used is called gel electrophoresis. Electro is inside the electric field. That means this process is done in the electric field. And phoresis is the movement. So movement in the electric field is called electrophoresis. And this is done with the help of the gel. So it is called gel electrophoresis. Now what we are doing for, yesterday we saw the DNA recombinant technology. If for constructing a recombinant DNA, we want the piece of the DNA where our gene of interest is there. So to get that fragment of DNA, we need to separate the DNA fragments. And that separation is done by this process which is called gel electrophoresis. Now the fragments obtained after cutting with restriction enzymes are separated by using gel electrophoresis. After cutting by the restriction enzymes, the endonucleases and exonucleases, when we are using endonucleases, they will cut the DNA at the specific recognition sequence. So once they have cut the DNA at specific recognition sequence, these DNA fragments, we need to separate them. And that separation is done by gel electrophoresis. Now in this gel electrophoresis, electro is we are using the electric field. So here electric field is applied to the electrophoresis matrix. What is matrix? Matrix is the medium. And generally the medium which is used here is agarose gel. Agarose gel is a natural polymer which we get from the seaweeds. So this gel is mostly used, commonly used for as a matrix for electrophoresis. And this agarose is the gel which is a natural polymer which we are getting from seaweeds. And negatively, the DNA fragments are negatively charged. Here we are using that characteristic property of DNA because DNA in the backbone, they are having phosphates, and these phosphates are having negative charge on them. So overall the DNAs are negatively charged. So when these negatively charged DNA fragments are put into the electric field, they will move towards the anode. So this characteristic property of DNA fragment is utilized here in gel electrophoresis. So fragments separate according to their size. Now here the separation is done as per the size. The smaller the fragment, the farther it will go. That means we will separate the larger segments, then medium fragments, and then the smaller segments. So we can separate the DNA fragments as per their size. So fragments separate according to their size by the sieving properties of agarose gel. Now this agarose gel is there to sieve it, to sieve these fragments. So the smaller the fragment, farther it moves. You will go at the end, you will be having very small particles. Now staining, which stain we are using, because without stain we will not be able to see the DNA fragments. So we have to use a stain here, a dye here, and that staining dye which is used for gel electrophoresis is ethidium bromide. So ethidium bromide is the dye, is the staining dye which is used to see, because otherwise we cannot see those fragments in the agarose gel. To see those fragments in the agarose gel, we are using the staining dye, which is the staining dye which is used is ethidium bromide. And then these fragments, they are not visible under the visible light. So the light also we have to use ultraviolet radiations. So these, after staining the DNA fragments with ethidium bromide, we have to expose them. The exposure to the UV radiations are used to visualize the DNA fragments. So two things are required to visualize the DNA fragment. One is the staining dye ethidium bromide, and the second is ultraviolet radiations, because under visible light these DNA are not visible. Now, DNA fragments are visible as bright orange colored blobs. Once you have stained them with ethidium bromide and then you have exposed them in the ultraviolet radiation, they can be seen as bright orange colored bands in the agarose matrix. Now, these bands are cut from the agarose gel. Now, once they have been separated in the agarose gel, then we can cut them, because gel is what? Gel is a very fluid type of a medium. So we can just cut it. It's not very hard. So we can just cut the gel and extract the gel piece. Gel piece means we can extract the DNA fragment which is present in that. And this process is called elution. So elution is the process by which we are extracting the gel piece where our DNA fragment is present. Now, DNA fragments are purified, and these purified DNA fragments are used in constructing recombinant DNAs. Because we know that for constructing recombinant DNAs, we require the fragment of the DNA, which is having the gene of interest. So once these fragments have been separated by gel electrophoresis as per their sizes in the electric field, then we can, after the staining and the exposure to the UV radiations, we can see them as bright orange color bands. And these bright orange color bands can be cut from the agarose gel and they can be extracted. And this process is called elution. And after that, these DNA fragments are purified and then they can be used for construction of recombinant DNA. Gel electrophoresis separates DNA fragments by size in a solid support medium, which is agarose gel. The rate of migration is proportional to size. Migration is movement. So the rate of movement of DNA fragments is proportional to size. The smaller fragments will move quickly and they will move farther. And the bigger fragments will move slowly and they will not move to larger distances. So smaller fragments move more quickly and wind up at the bottom of the gel. The DNA is visualized by including in the gel an intercalcating ethidium bromide. Now see this diagram. Electrophoresis definition is migration of charged particles or molecules under the influence of electric current. Literally, the electro means electric field and phoresis means transportation in under the process and in the electric field. So transportation in the electric field is electrophoresis. Now, what is the principle? The two anodes are there, a cathode and anode. Two electrodes are there, cathode and anode. So what happens whenever there is any charged particle in it, when we will put the electric supply on, the negative will move towards the anode and the positive will move towards the cathode. This is no electric field. When there is no electric field, they all are in the medium. Any charged ion or molecule migrates when placed in an electric field. The rate of migration depends upon its net charge size. What all things on which it depends? The movement, charge, net charge, the size, shape, and the applied electric current. So these are the four things on which these movement depends. So this is these are the two electrodes, and once this is on, the electric field is on, the electricity is passed, then what will happen? The negative charged ions or molecules will move towards anode, and the positively charged ions or molecules will move towards the cathode. So this is what is happening here. Here the medium is agarose gel, and in agarose gel, when the electric field is given, the electricity is applied, the movement of DNA molecules, DNA fragments are towards the anode because they are negatively charged. Now just see this diagram. This is the phosphate. The phosphate is having negative charge. So what will happen? The negative charges on DNA, which originates from the phosphate of the backbones. Now positioning DNA between negative and positive electric fields, as shown to the left, will cause migration. So this will cause migration, and these fragments will move towards the anode. Now this is how this instrument look like. You will find gel electrophoresis device, and you will find these are called wells. These wells are there, and from these wells the DNA is loaded inside the agarose gel. So here we will put the agarose gel. See this. This is the power supply. This is cathode, this is anode. Towards the cathode, we have the wells. And through these wells means the holes through which we can put the sample. And what is the sample? DNA sample. So this is the sample with us. In DNA fingerprinting, we were using this only, and we were putting all 2-3 samples, whatever we get from the crime scene, we put the DNA and then we compare. So here, this is the DNA sample. DNA sample is loaded through these wells, and this is agarose gel. So agarose gel is here. This is the cathode side from where you will put the sample, DNA sample, and this is anode. Now what will happen once you have put the sample and you have switched on the power supply? These fragments will start moving. So they will start moving as per their sizes. The bigger, higher molecular weight species will remain here only. They cannot move to larger distances. They will remain at the top. And the low molecular weight analysis, that means the low molecular weight species, the smaller fragments, they will move farther. So we can separate the DNA fragments. The bigger will be here and the smaller will be here. See another diagram. Now here also we have what you are using is power supply is there, and these are the wells. This is cathode and this is anode. And when the electric field is on, this is agarose gel, and this is the instrument to load the sample. So this is DNA sample loading. And once the DNA sample is loaded in the wells, these are called wells. So in these wells, once you have loaded, so these fragments will get separated. This is the fragments of this DNA sample. Then this, you can put different samples. And you have to use UV rays, UV radiation you have to use to visualize, because under visible light, they are not visible. So this UV rays are required for exposure to UV rays are required to make the DNA fragments visible to us. So we are using ethidium bromide and UV rays to make these DNA fragments visible to us. Now see this. This is mixture of DNA molecules of different sizes. So this is loading. This is the power source. cathode and anode. This is the gel, and slowly and slowly they will move. They will move as, and they will get separated as per their sizes. So once they have got separated, then we can remove them, and that is called elution. We can remove the cut the agarose gel, and we can take out the fragment after and purify it, and then use it for the construction of recombinant DNA. See, ultimately it will look like this. Now here you cannot see the colors, otherwise these are bright orange colors. When we see it under the ultraviolet radiation, we see these bands as bright orange colored bands. So these will look bright orange color bands. Here, what here you can see just the glowing thing. This is in orange. The orange bands are seen of these DNA fragments. So the DNA fragments now have got separated after this process of gel electrophoresis.